Practice 9. Plasmid extraction by the method of alkaline lysis.
Introduction. The alkaline lysis method is based on the use of strong salts, like sodium hydroxide. This is to lysate the bacteria, and then a neutralization has to be done in order to manipulate the plasmidic DNA. Although the procedure is quite long, the efficiency of such is considerably high, putting this method in the same category of the other methods like physical lysis or chemical lysis detergents. By using this sodium hydroxide, little pores in the membrane of the bacteria are made. Just as in those other methods are variations, the alkaline lysis also has variations, known as mini-prep and the mini-prep with STET buffer, that uses detergents and enzymes known as lysozyme to degrade the cell wall. Despite the efficiency of the three variants, they are all used on experiments depending on the aim of such.
Methodology. Note: all the centrifugations of this methodology need to be at maximum speed. As a first step, centrifugate 1.5 milliliters of culture for 20 seconds and remove the supernatant by decantation. Wash the pellet with 500 microliters of STE buffer and centrifugate it, and remove the supernatant. Resuspend the pellet in 100 microliters of GTE solution and 2 mg per milliliter of lysozyme, and mixing by vortex. Add 200 microliters of SDS alkaline solution and quickly mix 5 times and put 5 minutes on ice. Add 150 microliters of high salt solution and mix by vortex and place on ice 5 minutes. Centrifugate and transfer supernatant to another tube. Add phenol and chloroform in equivalent volume to the supernatant obtained and mix by vortex. Centrifugate 1 minute and transfer in aqueous phase to another tube. Add 2 volumes of cold absolute ethanol. Mix gently by inversion and incubate for 5 minutes at room temperature. Make another centrifugation 15 minutes in a temperature of 4 degrees Celsius and eliminate the supernatant. Add 1 milliliter of 70% ethanol and centrifugate 2 minutes in a temperature of 4 degrees Celsius and eliminate the supernatant. Lay dry the pellet. Dissolve the pellet on molecular biology water, and finally make electrophoresis to 0.8% of agarose using buffer TAE1X.
Results. The electrophoresis was made in agarose gel at 0.8% and TAE. In the first well is the DNA ladder oxygen 1 kV, 4 microliters, and from 2 to 7 are the samples for all the teams, but in this case we will be focused in the 6th well. Discussion. The alkaline lysis has many advantages as other plasmid recuperation methods, as simplicity, low cost, and reproducibility. And after that, it's necessary to make an electrophoresis with the objective to verify the quality of the samples and to see the visible isoforms of the plasmids. In the electrophoresis result can be distinguished the separation of the three isoforms, but as sharpness was milled, it could be by the incorrect addition of the sample in the well. Also, in the image is visible the contamination of RNA in the bottom, and the degradation in the movement of the samples in the agarose gel. There are any visible contamination by proteins, because the wells are empty in the image and everything was migrated. There were some optional steps in the protocol that was omitted, so if in the practice was made those steps, the quality and the visibility in the electrophoresis could be better, and also following step by step the protocol, taking care the quantity of chemicals addition and the time in specific temperatures.
Conclusion. It is important to state that when performing an extraction, the nature of the bacteria and the plasmid will be the ones that state the characteristics in order to choose the extraction method. It is worth mentioning that the neutralization step is crucial for when, by trying to visualize the results with an electrophoresis gel, the outcome will be unsatisfying, for the salts interfere with electrical current and therefore the visualization itself. However, all in all, the general outcome of the experiment was satisfactory.